Welcome to Lecture 6 in DIY transcriptomics. In the last lecture, we really dove in and spent the entirety of the lecture working in this step 1 script, and we accomplished two important things. The first is we used the TX import package to read our Callisto outputs into the R environment. And the second thing is we talked a lot about annotations, what it means to annotate data, and how we access annotation information. In our case, we used EnsembleDB and our human organism specific database package in R and Bioconductor to attach gene symbols to our transcript level data from Callisto. And at the same time as we read our data in, we took advantage of that transcript to gene mapping to collapse our data to gene level. But we also talked a bit about what to do if you weren't working with human, mouse, rat. What if you had, we're working with horse or dog or cow, and I actually used dog as an example of how to access annotation, a much wider range of annotation data. And so we used the Biomart package and we annotated a dog data set as an example. All right, so that's where we left off at the end of step 1, but we didn't actually talk about what we produced. We didn't look at the outcome of that step 1 script. So today we'll start where we left off. We'll actually open the step 1 script and just sort of talk about what it is we produced before we jump into step 2, and that'll really help us set the stage for understanding the transition there to the step 2 script. And so today, after we talk about what we produced, we'll move into the next step, the step 2 script, and we'll spend the entirety of the lecture in the step 2 script, and we'll finish that step 2 script today. And we'll accomplish a couple important things here as well. We will use some base R functions to filter our data, and then we'll use edge R to normalize our data. And I would love it if we could keep it that simple, but unfortunately, anytime we change our data in any way, or perform a process on it like filtering or normalization, we want to appreciate the impact that process had on our data. And so this is a really good opportunity to tap into two of the real strengths of working in R: wrangling your data and plotting it or graphing it. What do I mean by wrangling? I mean, we're talking primarily in RNA-seq data analysis about rectangular data structures, spreadsheets, right? Either data matrices or data frames. And so we want to be able to have a handle on those data types, to reformat them so that they're easier to mine and plot, to select certain columns or rows, and that's what I mean by data wrangling. For plotting, you understand what plotting is, but today, at the end of today's lecture, you'll understand how to use one of the workhorse R packages, ggplot, for working in this space, for graphing your data. As a reminder, there's really two on-ramps here into this workflow. The first was you downloaded the raw FASTQ files from the data page on the course website, and you created an index from a reference transcriptome using Callisto, and then you mapped your reads against that index using the Callisto quant function to produce these summarized reads. If you couldn't do that, either because you couldn't accommodate these FASTQ files on your hard drive, or for some reason Callisto wouldn't install or wasn't working for you, we provided a second on-ramp, which was we did the Callisto mapping and provide you the Callisto outputs so that you can just download those. And since they're simple text files, it's easy to just grab those from our course website and take off. So hopefully you find yourself somewhere in this workflow, and my hope is you can move quickly now that we're over this sort of awkward hump at the beginning of working in the command line in a terminal, and we're just going to be in R now, that you'll be able to pick up the pace and catch up, because our pace for the course will pick up. So I want to make sure you all do as good of a job as possible of catching up in the course thus far. Okay, so as usual, if you click on the lecture page for today, we can take a look at sort of what our goals are. The first thing to point out is there are some slides for today. It's not a lecture per se. It's more to help break up the monotony of looking at code in RStudio. But it also will help provide some context for some of the commands, functions, programs that we'll be using in the step 2 script today. Your homework is also due today. This is the second homework assignment, so it's important that you have a grasp of this tidyverse that we'll talk about today. So this is good timing to finish this homework, and we'll continue to use tools from this tidyverse in future lectures as well. Okay, so learning objectives. We want to briefly, as I mentioned, briefly review the essentials code chunk from the step 1 script. That helps us just sort of get our bearings and set the stage for going into step 2. We'll then start and finish the step 2 script today, and in the process of doing so, we'll understand the concept of a layered grammar of graphics and how we use this grammar of graphics, or GG, in a package called ggplot2. We'll then discuss the basics of tidy versus messy data and how we use the tidier package to go from messy data to tidy data, to convert messy data to tidy. We'll then filter our data to remove lowly expressed genes and then normalize our data to really allow us down the road to do these sort of important pairwise between sample or between condition comparisons. If you have not watched the lecture on units for RNA-seq, this is really where this turns from concept to practice. The idea of transcripts per million, of within and between sample normalization, those are the concepts we talked about earlier in the course that now become an essential part of our practice in the script. And then, of course, you'll be working with the step 2 script, which you can download here or on the code section of the website. Okay, if that sounds like a lot, it's because it is. And frankly, I find this lecture one of the hardest to give. And on your end, it's probably one of the harder to sort of digest all the different parts of this lecture. And that's really because we're trying to live in two worlds here, or wear two hats. One is we're trying to do bioinformatics with RNA-seq data and filter and normalize and several other things. But in order to do those things well, we also have to put on our data scientist hat and do plotting, care about the structure of our data, and understand how to slice it in different ways. Okay, so at this point, I think we can, leave the slides, go open the step 1 script, and review this essentials chunk. So I'm going to head over to my file browser here, and just, I'm in my working directory. I'm going to double click that R project, because again, that allows me to pick up right where I left off. I'm not going to open the R scripts. I'm going to open the R project. And just as I had hoped, we pick up right where we left off from the last class. The step 1 script is open, and I can see the objects in my working environment are the objects that I created in the last lecture when I ran this step 1 script. So I'm going to just scroll down to the bottom of the script, and remember I can do that using my handy table of contents here. I can scroll down and go to the essentials, and I'm going to go to the essentials because remember that code chunk provides only the essential lines of code needed to reproduce that step script. And I can see that step 1, for as much time as we spent in it, really can be reduced to 7 lines of code, right? These 7 here, and 4 R packages from our library, and that's it. So I'm going to run each line, one line at a time, and as I do that, I'll kind of just verbally annotate this so that we have our bearings on what it is we actually did in step 1. I'm going to load the tidyverse package. As I do that in the console, I'm going to see some information here. Remember this, I will come back to this in a slide in just a minute. We're going to load tx import, which we used for getting our Callisto outputs into R. I'm going to load Ensemble DB and our organism-specific database package. Those were both used just to get annotation information, gene symbols, to attach to our transcript, Ensemble transcript IDs that were in our reference file that we mapped our data to. That's ready, so I've loaded those 4 packages. I'm now going to start moving through these 7 lines of code, reading in my study design file, and that'll be a data frame, of course, reading in a set of file paths that I used the column, one of the columns from my study design file, and just attached this character to it to create file paths. And then I'm going to retrieve some information from my database package, my Ensemble DB Homo sapiens database package. I'm going to get transcript IDs and gene names. So I do that using the transcripts function. And I'm going to convert that into something funny called a tibble, which is just short for a tidy table. It's just a data frame, but it's a data frame that will display a little nicer in my console and that will play a little nicer with some other R packages. I'm now going to use dplyer's rename and select to rename one of these columns and then select two of the columns from my initial TX data frame. This effectively gives me a one-to-one, or a many-to-one rather, mapping of transcripts to gene symbols. And I can use that now in my last part of the step 1 script, where I use TX import to go to those file paths I set up, find the Callisto outputs, recognize that they are in fact Callisto outputs, so type equals Callisto, recognize I have this transcript to gene mapping in TX, and read my data into the R environment. So I'm going to do that now. It's going to read all 10 files and we'll be good to go. As a quick reminder here, one important thing you may want to play around with is I have TX out set to false, meaning I do not want transcript level data, TX, output when I produce this object. And that's because I want to use this transcript level data to give me gene level expression data. You may want to, from time to time, depending on your interest, you may want to change this to true. That would set this up to produce transcript level data. If I do that, I could change this to transcript so that you don't overwrite your TXI gene object, and I could basically produce this second object, right? You don't even have to choose. You can make them both, TXI gene and TXI transcript. All right. The first thing I wanted to point out, we're now at the point where you now understand what we did in step 1, but what did we actually make? Well, we made these two objects, or one object really, TXI gene. I just now did this sort of ad-libbing here, making TXI transcript. But regardless, they're just lists. They're large lists, but they're lists. What is a list, right? This should be familiar from your homework, but I think it's worth just quickly reminding you that there are these different data types in R, and we'll be working across all of them. The simplest is a vector, which is just a series of elements in this sort of single entity. This could be a numeric vector, where each of these blocks is a number. It could be a character vector, where each of these is a letter or a word. So every word in my lecture today is just a really long, probably too long of a character vector. It could just be the alphabet. You get the idea. A matrix is comprised of rows and columns. And in particular, a matrix in R is comprised of the same kind of data. All these blocks are the same color. And for our purposes, a matrix with RNA-seq data is just numeric data. The only caveat here is that a data matrix in R has this special case of row names that are considered separate and distinct from the data itself. So although a matrix has to have only one data type, and in our case numeric data, it can still have gene symbols as row names. All right. An array is just several matrices sort of stacked together. A data frame is very similar to a data matrix, rows and columns, but it can have a mix of different types of data. It could have a column that's numeric, a column that's characters. It could have a column that's dates. And then lastly, and relevant to what we just talked about, is R also has this idea of a list, which is just any combination of any of these previous data types. In the case that's shown here, this list has 6 elements. Each of these things is called an element, and those elements are 3 vectors, a matrix, another vector, and a data frame. Okay, so if we go back to RStudio, we now have a better idea of what this list thing is, and if we click on one of these, we actually see the elements in the list. It's got 3 different data matrices. Each of these is really large, 35,000 rows by 10 columns. I can see it's all numeric data, just seeing the values over here. And then it's got a single character vector of length 1. And I can see the length of the character vector here. And it's length 1 because it literally consists of a single word, length scale TPM. And that's just capturing from what I instructed it to do here. All right. Okay, so now you understand what's in TXI gene, what TX import actually created, and it's just a list. And that list is primarily just a set of different data matrices. If I click on TXI transcript, I'll see the same thing. It's a list, but it's a lot bigger. It's 187,000 rows by 10 columns. So way bigger. If you take, by the way, the total number of rows and multiply it by the number of columns, you'll get the number of total elements in the list as well. It's not so important. But two different lists that really vary depending on whether I read my data in at the gene level or the transcript level. Okay, so that's what we created at the end of the step 1 script. Now let's actually go ahead and take a look at that data in more detail, but do it in the step 2 script. So I've already downloaded this step 2 script from the website. You should do that now if you haven't already. Drop it into your working directory and you should see it in the file browser and just click on it. Now I'm looking at the step 2 script. I can leave this step 1 script open. You can have multiple tabs in your text editor here in RStudio. And we're going to do a few basic things that we just have to get into the habit of doing every time we open a script. The first thing is that we want to go to Edit, Folding, Collapse All, so that we can really see this high-level view structure of the script so we can understand what we're trying to do. As always, the script starts with two code chunks that are standard, an introduction, and loading the packages. But then we do a few things here that you're probably already familiar with from my intro. We're going to take a look at the data. We're going to generate summary stats for our data. We'll create our first plot. We'll then make something called a digital gene expression list, so another list data type, but one particular to dealing with RNA-seq data. And we'll use that to create some plots as well. We'll then filter, normalize, and then we just have everything distilled down into the essentials as usual. So that last code chunk is standard as well. All right, I'm not going to go over the intro because it's stuff we've already discussed. The load packages, we've discussed loading packages, downloading and loading packages already. I'm not going to go through that again, but I'll just tell you at this point, this is where you could pause the video and go and get these packages, right? You already have the tidyverse if you ran the step 1 script, but you don't have edgeR, you don't have matrix stats, and you don't have cowplot. So you can use the install on the packages tab to go ahead and go get those now. And remember, set repositories as a function will change this from just looking at CRAN as the default repository to looking at, I suggest just setting all of them as default so that you make sure you can get these. We'll use edgeR, which is a really well-known package for differential gene expression analysis, but we'll only use it for one function, one or two functions, which is sort of dealing with and creating these digital gene expression list objects. We won't actually use it for what it's designed for, which is differential gene expression analysis. And this is just a personal preference of mine. I use Lima. It's what I learned to use first, and so it's what I prefer. But edgeR, DESeq2, and Lima are all great. They're all well known for their ability to do, sort of broadly speaking, differential feature selection. So in other words, not just finding transcripts or genes that are different between two or more conditions, but finding any features that are different in a data set. And so they've been applied, for example, to finding bacteria taxa that are different in microbiome studies. We'll use the matrix stats package to calculate, it's really simple, to calculate stats on rows and columns of a data matrix. And not essential, but we use cowplot to kind of, take any plots, multiple plots, and throw them together in a single figure. And by throw them together, I mean put them together in really nice ways that really help you get very close to producing a final figure just in R. Okay, so I'm going to go ahead and let you download those. I already have them in my library, obviously. So at this point, I'm going to load those packages. Now, this is a good time to remind you that in the step 1 script, when I loaded the tidyverse, I got this whole sort of blurb that played out here in the console. And I told you we'd come back to that. And now is as good a time as any to talk about this. That's what we saw in the console. We saw when we loaded the tidyverse, we talked actually in a previous lecture about this idea of conflicts and how generic function names like filter, select, could be the names of functions from multiple packages. And so dplyr does some work here to tell you, hey, we're going to enforce that if you use filter, it's going to come from dplyr. It masks the filter function from the stats package. So we already talked about that, but I wanted to now take this opportunity to highlight that when you load the tidyverse, even though you're loading it as if it's one package, it's actually 8 and arguably 9 different packages that make up this universe, tidyverse. And they become such a fundamental part of what we're going to do in the course and what many people do in R that it merits spending a little bit of time talking about it. And I think this is also a great time to just introduce what might be kind of a fun or silly topic, but I really like it and many people do, which is it's kind of boring to look at package names. It's much more exciting to look at pictures, and if you're more of a visual person like me, you might really appreciate that there are icons or badges that are associated with each of these packages. And this is beyond just the tidyverse. This has become a real popular trend in R and Bioconductor, which is creating these so-called hex stickers that have, relationship to the packages. So there's one for every package or for most packages. And they almost act as like merit badges now, where once people use a package and they feel like they either like what that R package does or have some level of competency in it, they'll decorate their laptop or their, desk or their office window with these stickers. But it also provides a way for us to kind of couch some of the functions we've already used and some of the functions we'll be using ahead. This is also a good time to just say that the brains behind these packages is a very famous statistician and R programmer named Hadley Wickham. He was initially faculty at Rice University and is now one of the chief scientists at RStudio. And he is the original developer of ggplot2, a really just critically important package in R. dplyr and the whole tidyverse, and he and his team have continued to develop and enhance and build upon this ecosystem of data science tools. He is such a dedicated advocate of open science. Obviously before even moving to RStudio, he was creating open source software packages that were really heavily used. So it's again an example of how, even though RStudio is a commercial entity, a lot of the key people there come from an open software and open resource background. He's so dedicated to the R environment that he even signs his name as an R function. So this is actually a picture from the book Camilla had signed when we were both at the RStudio conference, and of course Hadley was there and signed her book as book, parentheses, quotes, R for data science. That's Hadley's book. SIG, parentheses, his name in quotes, and then each time he's piping it, which we haven't talked about yet, but here's the pipe function here, 4 quotes Camilla, right? So this guy is just completely immersed in our culture, and it's great. And it's great for us because he's developed these fantastic resources. Okay, so I told you we'd use these hex stickers to kind of couch some of the things that we've talked about. You actually have already used several of these R packages from the tidyverse. So you used the read TSV from Reader when you read your study design file as a data frame into R. You used the Tibble package when you used as Tibble to convert this TX object of annotation data that you pulled out of EnsembleDB package when you pulled that data in using as Tibble. You use the rename and select functions from dplyr, and you will, but you haven't yet, use the ggplot function, the main function from ggplot2. And we'll also dig into what I think is one of the more complex and sophisticated packages in the tidyverse called tidyr. We'll just barely scratch the surface here. We're going to use the pivot longer function later in the lecture for sort of changing the format of some of our data. We're not going to use per, stringer, or for cats, a package for dealing with categorical variables, not actual cats. But we will use this magrittr package, which often is left out of the sort of tidyverse spectrum because it really does one thing, which is provide the ability to use this % greater than % pipe function that we can string together code in a really efficient way with. Okay, so that's the idea of the tidyverse. So now let's sort of toggle back to RStudio. So you understand the tidyverse. We've loaded these other packages, and now we're ready to actually start working with the data that we read in step 1. So as an example, the first thing to highlight is that any list object, which we now understand TXIgene is a list, any list object can be accessed using that same dollar operator that we used for accessing data frames. Recall, data frames, we can tap into any column by typing the $, and RStudio will actually list the columns and we can select which one we want. Similarly, when we have a list object we're dealing with, if we type the $, RStudio lists all the elements in that list. So we know there are 3 data matrices and a character vector of length 1. Here they all are. So if I select the first one, abundance, this is that abundance column that was in each of your outputs from Callisto. It is the transcript per million measurements for each transcript or gene, depending on what you read in. And so I'm going to take the TXIgene object, $abundance, and read that into an object called myTPM. All right. When I do that, now I can see I have a large matrix with 356,000 elements. 356,000, why that many elements? Well, if I just click on that, it'll open in my data browser and I can see gene symbols. Remember, row names in a data matrix are not considered part of the data. The rest is numeric, so it's a data matrix, and it has 35,000 rows, 10 columns. You multiply those two numbers together, you get this number of elements. So the number of elements is the number of data points in the data matrix. If I change that from TXIgene to TXI transcript, that's going to be a much bigger spreadsheet, and I will change the name of the object just to avoid any issues. And now here I have a large matrix with almost 2 million elements in it. If I click on that, even though this is a huge spreadsheet, it has no problems opening in the data browser. I see, of course, now no gene symbols because this is reading the data in at the transcript level. So I have transcript IDs. And I can see it's 187,000 rows plus long and still 10 columns. A lot of transcripts here that have really no evidence of expression, at least in my data. But if you scroll down, of course, you're going to see evidence for expression for many of these. Okay. And if you recall, one of the advantages of using TX import is that you can take these multiple transcripts per gene and use those multiple measurements to get a more accurate assessment of gene level expression, which is why I like to read the data in at the gene level. All right, so I'm just going to undo those changes I made to the code. So that's the TPMs, but there's another slot or another element in that TXIgene object called counts. And we talked about this. Counts are the actual number of reads mapping to that gene in that sample. And so if I take that element or that element of my list and I capture that, I'm going to put it in an object called myCounts. All right, so I have my transcripts per million, I have my counts. Now I can use the column sums function, call sums. This is a base R function. I can use this to tally up the total for the transcripts per million or the counts in each of the 10 columns, each of the 10 samples. So if I was giving this lecture live, this is the point at where I would pause and sort of push you with some awkward silence to predict or guess what you think these tallies might come to. But awkward silence does not go over as well on video. So I'm going to just run this and we'll talk about what we see. So call sums of myTPM, if you think about what that is, it should be 1 million, right? We talk about each transcript, being some proportion, right? It's relative measurement, proportion of 1 million. So if I reach into a cell and I sample 1 million transcripts, I know how many of the transcripts out of that 1 million will be gene X. And you can see these numbers all are close to 1 million, but not quite, and they're not all the same. And I think that's a function of the fact that transcripts per million is really meant to refer to transcripts, not genes. And we sort of changed our data, right? We read it in and collapsed it to gene level. So if instead of looking at the myTPM object, I looked at my transcript, myTPM at the transcript level, and I did call sums, that should absolutely total to 1 million for each column. And indeed it does, right? No differences. It's all 1 million all the way across the board. So something happens when you go from transcript level data to gene level data that they just don't sum to 1 million. It's close, but it's not exactly it. All right. So in a similar fashion, we can talk about what we should get when we tally up the counts. And if I run that, I see really large numbers here, 45 million, 55 million, 74 million. These should look familiar. Hopefully these look familiar to you. These are the total number of reads mapping to my reference for each sample. All right, so that by definition, if 74 million reads mapped from that sample to the reference, when I tally up all the reads mapped for all the genes in that sample, it should come up to that number. There shouldn't be anything missing because we mapped against that reference. So that's how to relate to these two data matrices that we pulled out of that TXI gene list object, and how it'll differ if you dealt with gene level or transcript level. Okay, the last thing that we captured in that step 1 script was the study design file. We called it targets. We don't need to spend any time on this today. Just to remind you, it's a tibble. And there's very minimal information about these samples. We know whether they were healthy or whether they were from a patient with cutaneous leishmaniasis. We have some sample label, and we have an SRA accession from the short read archive, a public where we can go to find this data in a public database. As a quick reminder, whatever you're doing with your own studies, I would encourage you to have a study design file that has as much information as you have about each sample, mouse, patient, whatever, or about the actual wet lab work. Were the first 2 or 3 samples prepared on one day for the libraries and the others prepared on a separate day? In other words, was there a batch effect or a batching of your samples? Those are the kinds of information that if they're in your study design file, you can actually use it to ask questions or interrogate your data down the road. One last thing I like to do is to dip into this targets object and grab the column that corresponds to what I think constitute good sample labels or sample names. Why is this useful? Because there's going to be lots of instances moving through this script where we'll want to throw sample labels on a graph or on a spreadsheet, and it's nice to just have them in in an object that's logically named. So I'm going to grab, again, because this is a data frame, the dollar operator becomes really useful for grabbing any column. I'm going to grab $sample and put that into sample capital L labels, so that now I have a character vector of length 10, where each element in that vector is one of the sample labels. All right, so that's a good point to stop and stretch your arms and legs and rest, and then we will pick up with the rest of this in the second half of the lecture.